It's a really a great pleasure for me to introduce Dr. David Relman to you today. I have a whole bunch of things to say, so I'm not sure I'm going to get through all of these things. I think the take-home from my intro is that David has had a very interesting and varied career. He started off getting a bachelor's degree in biology at MIT. He then went on to Harvard Medical School for an MD and did clinical training at Mass General. And from there, he moved out to the West Coast, to Stanford, and I think you did a postdoc with Stan Falco. Stan Falco was a giant in microbiology. Stan passed away last December. So I think a lot of the things that have stimulated David was certainly sparked by the initial interactions and I think continued interactions with Stan Falco. So David has been to Purdue. This is his third time. So this is a joint invitation from Discovery Park and Tomas and Pi4D and myself. And it arose out of his previous visit here at Purdue, which was for some type of security conference that Tomas and Discovery Park ran. And at that time, I was kind of surprised. Of course, I was out of town, and so I couldn't participate. And I was kind of annoyed not knowing about it in advance because David and I have been on an external advisory committee where we meet in Hawaii every year for the last few years. And so I knew David, and so it was so disappointing to have him come to Purdue when I was not here. But he was here 3 times, and so the time before, which he told me about last night, I quickly realized that I was not there for that visit either. That visit was in 2008, and it was spurred on, I guess, by then presidential contender Barack Obama, and he was hosting was it a biosecurity roundtable here at Purdue. It was Senator Obama and former Senator Evan Bayh was brought in as a co-moderator, and there were 6 panelists that included David, and so David had an early opportunity to interact with our former president. And I don't know if David's going to mention it, but President Obama had some interesting questions for him. David has, to distill it very, very quickly, has had 2 kind of major thrusts that have taken him in different, but in some places overlapping intersections. I think his early history was in pathogen discovery. And one of the interesting tales that he could tell, I don't know if he would tell, was he was involved in a National Academy's committee that was looking into the anthrax incidents, the mailing of anthrax in envelopes to a variety of people back in 2001, yes. And David and the committee that he was working with were looking into how the FBI conducted the investigation, which eventually named a potential suspect for those anthrax mailings. He has, in kind of part 2 of his research portfolio, so that kind of is in the area of pathogen discovery that led him in that direction. And the second part has been in studies in the microbiome, and David has been one of the pioneers in looking at the microbiome, and really with an emphasis on the human oral and gut microbiome. And so I think we're going to hear little bits about this today. He is a fellow of the American the American Academy of Microbiology. I should know that because I think I'm also a fellow of that. And he is also a member of the National Academy of Medicine. He has been on too many committees to mention. one that he is currently on is the Intelligence Community Studies Board of the National Academies. And previously, he was on the Lawrence Livermore Advisory Board for Science, Technology, and Engineering. And I think you were a member of that board for 11 years. And that is where he encountered Thomas. And so there is a connection there. So I think he's going to give us a very varied overview of some of the topics that are of interest to him. I think they're going to be of great interest to all of you. I think he's going to sprinkle that in with some pieces of career ideas for those of you. So David, welcome. I'll take this.
Thank you, Richard, for a very kind and generous introduction. So this is a daunting task because I know some of you and the good work that happens here. And in fact, the whole morning I've had is kind of a lesson in some really interesting ways of how to blend really different kinds of science and engineering together. So I know that so this audience is pretty diverse in its interests and backgrounds. That's challenge number one. Challenge number two is that if the topic is to be microbiome or microbiomes, well, that by itself is a huge, growing and a bit ill-defined topic. And so picking parts of both of those kinds of interests and backgrounds is difficult. So this is going to be necessarily sort of skewed and selective and not in any way comprehensive. And I also want to emphasize the work of others that have given rise to ideas that I think are interesting and maybe worthy of a broader conversation if we had time, which we probably won't. These are the things I thought I'd talk about, just remind you about what we know and don't know with respect to the benefits that we're associating today with the human microbiome, but think more broadly. These are benefits that are true in general form for many microbiomes and the environments in which they reside. And then a little bit about mechanisms that we think might be at play, but as you will hear and probably know, we're woefully short on understanding those. Then we're going to do a little bit of influence of disturbance ecology, starting with lighting and how it affects human biology and perception. And this is in one area in which I have some personal interests and laboratory interests in recent times. I'm just going to touch very briefly. This will not be a complete or even necessarily understandable story, but I hope to at least share some of the observations or the ways in which we're approaching these questions about the nature of disturbance and stability and resilience in complex ecosystems. I was asked if some of this could be made relevant to disease. Again, think broadly about what disease really means, pathology or altered states in which there seems to be harm being done by one party or both. And so I'm going to talk a little bit about a human disease that seems to be linked to the human microbiome, but just again, a little tidbit. And then talk about possible interventions and where that might lead both from a beneficial perspective, but also from a risk and misuse or malintended perspective as well. So that's a lot. I may have to go a little bit fast here. Just a word about history in case some of you think that this is a very recent topic that was just ginned up in the last few years. Microbiomes have been the subject of work and thinking for thousands, really hundreds and even thousands of years, although it was only until the late 1600s that humans became aware of exactly what was meant by a microbe or a microbial community, although at the time they were called little animals, but it was made possible through the invention of the microscope. So the lens of a microscope was the first lens through which one saw the microbial world. Von Leeuwenhoek looked at the scrapings from human teeth, among many other materials and made the first descriptions of the human microbiome. Second lens was that afforded by cultivation, the means to propagate organisms that we can't see so that we can see them as a collective or the products of their activities in easier formats. And that led to a huge amount of focus and insight into the microbial world, focused largely on pathogens, on disease-causing organisms, but there was a lot of environmental microbiology that was born in the early 1900s and led to some really interesting observations and challenges having to do with the fact that even now, with the ability to grow so much, it seemed that there still was a large fraction of unseen, uncounted microbial life because of the discrepancy between single cells that could be counted under a microscope and single colonies that could be counted on a plate. And then finally, the third lens, which is that of the modern molecular era and the use of molecules as readouts when chosen properly for ancestry and evolutionary relationships. Again, if you pick a molecule that has this clock-like, reliable clock-like behavior, then one now has a means of recovering information directly from an environment without reliance on cultivation or imaging. And now there's a third lens. And through that lens, this modern resurgence of interest in microbiomes has been fostered and born. And then there's this convergence. And it's very much what Discovery Park seems to be about as well. A lot could be said on this topic. I'm just going to show you one list, an incomplete list of some of the kinds of benefits that we now associate with our microbial communities, human microbial communities. Same is true for animals and in many ways other hosts, and indirectly also true of other environments. So food processing and digestion, the generation of nutrients and acquisition of nutrients, processing of foreign chemicals, foreign to the host, co-regulation of a variety of metabolic processes, development, etc. And all of these are really interesting stories that are only partially told and partially understood at the molecular level. But I just give you a few further insights into two of these. One of them is the role of the human microbiome in the early postnatal development and differentiation of the immune system. We know now that there is host species specificity to the effects that have been seen when germ-free hosts are repopulated with some relevant community. Much of the work that has been done with the reconstitution of colonized mice with mouse microbiotas has shown the recovery of a lot of otherwise deficient features in the immune system development. And in this work that Dennis Kasper spearheaded, the evidence was provided that if you put a mouse microbiota back into a mouse, you get much better recovery of mouse immune cell numbers and cell phenotypes than you do if you put a human gut community into a germ-free mouse, or even a rat microbiota into a mouse. And so this, again, is a general theme that has to do with the increasingly, seemingly exquisite tuning of communities to their particular host, not just at this host species level, but even at the host individual level to some degree. And that, of course, is one of the big challenges right now, is to understand the basis and nature of individuality within strains of hosts, like human individuals or other hosts. The other one I just wanted to mention was colonization resistance, the property of climax communities to exclude foreign would-be members from colonization and proper introduction into the community. Community membership, so the club has exclusive properties, and the question, this has been recognized actually for 50 or more years. This is 1962, and this is not the first, but one of the early observations that only when a wild-type mouse microbiota is knocked down in numbers and types with an antibiotic, can you now install or infect the mouse easily with an invasive outside pathogen, in this case, Salmonella? Or at least the numbers needed go down dramatically by several orders of magnitude with antibiotic treatment. So this kind of observation that suggests this property of a climax community has been known for some time, and now there is sort of an increasing effort to understand the basis for that. And a lot of it comes down to the products and small molecules that microbial communities, microbes as individuals, but microbial communities in particular, use to talk to each other, maintain their community structure, and also relate to their environment. So us, if it's a human microbiome. These are some of the small molecules. I understand that Michael Fischbach was here for the Microbiome Symposium earlier this year, so this is one of his slides, and you will have heard many stories that emanate from a starting point like this, where people say, how much of what circulates of a small molecule nature in, say, human serum and plasma is of microbial origin? And you start to come up with a lot of these structures that appear to be at least in part of microbial origin. So what are they doing? And might they be related to some of those benefits that I showed you on a previous slide? So now there are an increasing number of studies, mostly based in laboratory animal models. This is by a colleague, Denise Monac, who in this case narrows down and identifies propionate, a short-chain fatty acid made by gut microbiota, mostly in the colon, that has this property of actually altering Salmonella intracellular pH environment and altering the sort of the viability and biology of Salmonella. And this, it seems, in at least the models that she created, may play some role in preventing Salmonella from securing a foothold in an otherwise intact native wild-type community. But the story is always more complicated than a single small molecule. And so when people have looked at other potential invading organisms like Clostridium difficile, the story is slightly different. And it's not that propionate is not important, but here rather through a large kind of funneling effort, people arrive at secondary bile acids or other short-chain fatty acids as potentially important in excluding or inhibiting Clostridium difficile, another would-be pathogen if allowed to flourish in an otherwise filled niche. And then finally, this paper just from a few weeks ago from Eric Paymer's group suggesting that this very particular molecule, it's a lantibiotic by class and type, made by one of a number, but a discrete number, of commensals may have some particularly important role in excluding or inhibiting this would-be invader in a population of humans. Vancomycin-resistant enterococcus is a huge opportunist in the setting of an impaired immune system and or a heavily damaged microbiome previously subjected to multiple rounds of antibiotics. This was a population that he was very and has been very interested in, and this story kind of derives from observations not just in mice, but this panel at the bottom are actually data from humans that he went back to and asked, do I see the kinds of correlations I'd expect if this lantibiotic and its gene were truly associated with exclusion of this vancomycin-resistant enterococcus? And you see that on the left, there is a pretty good relationship between the abundance of the gene on the x-axis and the relative abundance of VRE on the y. But on the right, you see that it's, of course, more complicated because there are subjects in whom there seems to be very low levels. You can see on the left, low levels of antibiotic-producing genes, and yet still not much VRE, even when present in some number. And there, the correlation is with alpha diversity. So overall numbers and relative abundances of the community, which must be producing other things as yet to be identified. So those are the kinds of efforts that people are undertaking through that particular lens, that of small molecule chemistry and trying to explain some of these properties, but we are really, in my humble estimation, way at the beginning of what you would have thought we understood a whole lot better because of hundreds of years of very kind of circumstantial observations. So, and that is just the human side, which we, of course, always care about more than anyone else. It's us. It's all about me. If we really cared about the ecosystem, we'd care about the other side of the partnership. What is it that a good commensal requires, and what is it getting from this relationship? And this list is pretty simple sounding and looking, and that's because our understanding is still even more simple than the previous. So I want to switch to this topic of disturbance based on what I hope will be your willingness to agree with me that for now, the idea of a stable system that provides benefits is something that we ought to be able to understand, or we should be understanding, and a system that's lost some of those benefits to its host environment is one that we want to understand from the point of view of restoring back to the beneficial state. And here's where ecology has a lot to offer in the way of ideas. I thought I'd share some that you probably all understand well. And there are a lot of schematics that help people understand stable states in complex systems. So meteorologists and physicists and economists all use this sort of these depictions, although many of them prefer the mountain peaks as the points of optimization rather than a valley as stability, but I'm picking valleys here. And there are a couple simple points. Any system has multiple alternative stable states. They're not necessarily all favorable when you're viewing through a particular lens, like I care about this one benefit. Not all provide that benefit. The other thing is that it's the topology of the landscape that really matters here. If you want to be able to predict where someone is, or more importantly, how likely it is that their ecosystem will continue to stay in that state, then almost by definition, you have to understand that landscape topology. And this is where I think there's a lot that we could do to improve our understanding to predict and understand stability by thinking of experimental approaches to help us describe the topology of a current landscape in an ecosystem. And I'll explain what I mean by that. But I'll just tell you now that I think one important approach is deliberate, defined, standardized forms of disturbance. Because without disturbance, you often have no idea, for example, how deep is a valley, the kind of energy or factors might be needed to displace a community and get it to move to another. The other lesson from depictions like this is that topologies can change, and they're doing so all the time. As you sit here and get older, your immune system is senescing, although not dramatically during the course of this lecture, unless I take a really long time to get through. But in general, it is. And meanwhile, when you have decided to move to a new place with a different diet or food source or activity lifestyle, then the topology of your ecological landscape has changed again. And when you take an antibiotic, it certainly is. And when you suffer onset of a chronic inflammatory condition, it certainly is as well. So it's complicated. But disturbance has a pretty bad name these days in the lay press and even among members of our microbiome research community, but it should not have such a bad name for the reasons you've probably read by now. It's just a natural feature of all ecosystems, and it has served over long periods of time the good of an ecosystem in purging intercurrent invasive species, in resetting and allowing a system to reconstitute itself, usually with reservoir features that are the most important, that are the most preserved and protected. So it can be very helpful. But of course, we think of disturbance in human terms about things that we are doing to ecosystems around us, and those are kinds of disturbances that are usually of greater magnitude and or of greater frequency than the system in evolutionary time frames has ever had a chance to accommodate, and that's where you get into trouble. So far, what we understand about stability and the existence of alternative stable states has come from observational studies like this one, relatively few, in this case, human hosts, but they're outbred. I mean, they're living whatever lives they choose of a somewhat natural sort. And these two individuals, A and B, studied themselves over the course of a year. And in short, really interesting paper, and recorded everything about their life that they could. And where you see swatches of dark blue, it's generally because organisms that had been present in relatively greater abundance have now become depleted. And where you see orange, you see blooms. And you see two basic events here. Subject A in the gut and saliva had an event around here at day 100, and subject B later in the year. Those events map exactly to a move by subject A to a foreign country where he or she lived for more than a month, and you see pretty good recovery afterwards when they move back to where they came from as adults. And subject B became ill with a diarrheal illness and took an antibiotic. And here's where you see, a story that still hasn't been completely told one way or the other by the end of the study. In other words, there was a disturbance that continued, and there did not appear to be recovery. So that's the kind of thing that we have to work with. And yet, in the ecological literature, there's a great deal of interesting sort of instruction on how one goes about designing carefully executed disturbance studies. What's the theory and the design that would serve well? Although much of it is really based upon the macro ecological world. A lot of this work that we often refer to when we're trying to understand the microbial ecosystems is based upon for example, plant communities that have been studied, but counted in a very different way, of course. They're visible. And from which some interesting conclusions have been drawn. So each of these is supported by several studies over the last 50 years. And what comes out of an effort to answer the question, what correlates with resilience, recovery, are these sorts of things. Numbers of species, relative abundances, the nature and numbers and kinds of interactions, the presence of redundancy in the system, multiple species doing the same thing, so-called insurance hypothesis in ecology, and more. But none of these has proven to apply across many kinds of communities and forms of disturbance. So we undertook a somewhat ambitious study that's still ongoing, involving some human subjects and some deliberate forms of disturbance. Our goal was to derive composite features of the microbiome that seemed to predict or be associated with subsequent stability or resistance to a disturbance or to successful recovery from a disturbance. Because we hoped to derive common features to multiple forms of disturbance, we picked three of very different types. We picked one disturbance that we call the diet perturbation, and it was 5 days of Bob's Red Mill potato starch, 40 grams a day, so a resistant starch that subjects would not have been routinely eating. So it's a supplement to what was otherwise their usual diet, which we asked them to continue without other major alterations. The second disturbance was a physical purge of the ecosystem. We used a cathartic called Go Lightly. It's a polyethylene glycol-based solution that's used to do a colonic cleanout. Some of you, perhaps, I don't want to point fingers or anything, but I bet you some of you have had a colonoscopy. And if you have, then you've gone through this. This is about a 12-hour procedure. And we asked subjects to do that, but without the colonoscopy, just because. And then the third thing was 5 days of ciprofloxacin, a fluoroquinolone, 500 milligrams twice a day, 5 days, pretty short course of an antibiotic that was actually chosen because it's predicted to have relatively little anti-anaerobe activity, and we knew those would be the dominant organisms in the distal gut that we probably didn't want to mess with too much for fear of major both clinical and biological problems. Okay, so that's what we did, and we recruited. We actually thought we were going to have a problem getting the colon cleanout people, so we aimed for 40 and had 60 in the antibiotic arm. They each got the diet disturbance ahead of time. So the way this worked is daily sampling for a week, and then 10 weeks of observation with weekly sampling. Then it goes back down to daily. This is actually based upon statistical frameworks for understanding variance across data. So it's daily just leading up to the first disturbance, which in both cases is the resistant starch. Then it's daily and weekly again, 10 weeks, then daily, and then the second disturbance, either Cleanout or ciprofloxacin, and we had a no perturbation arm. Okay, guess where we had trouble recruiting? Yeah, so that was the surprise. We could not find, we had a hard time finding people to take Cipro for 5 days without needing it, even though most of their physicians were routinely prescribing antibiotics that they didn't need anyway. But this is where the microbiome literature now has actually come full circle. We started using Cipro in 2008 and had no trouble whatsoever until we started publishing our results. And so that led to a problem now, but we still got 60. Meanwhile, the cleanout I thought was going to be a horrible problem, and no problem whatsoever. Everybody wanted a cleanout. Seriously. People wanted to pay us to do this. Yeah. So we just now have all of, I didn't tell you, multiple kinds of measurements on lots of these samples. So from every sample we did a taxonomic profiling based upon amplified marker gene, 16S ribosomal RNA gene segments, amplicons, reliable, tells you in the way in which we compute and infer sequence types. It's often reliable to the strain level, but not always. Depends where you are on the tree you are. We also did sort of shotgun metagenomics on a subset of these samples, actually doing it on all. Metatranscriptome, so the transcript profiles across the microbial community for a subset of these. That's expensive, and metabolites from the same. And the idea was merge data using sort of modern multi-table methods and infer features that are correlated with these important phenotypes. So we're still in the midst of that. I'm just going to show you, again, little tidbits because I know we don't really have time, but these are all of the 16S-based amplicon data for the 3 disturbances, and you can see right away that one of them had a much greater effect and a longer lasting effect than the other 2. And that on the left is the antibiotic, ciprofloxacin. Middle is the diet disturbance, and the right is the colon cleanout. And there's a lot that could be said about each of these 3 stories. I chose not to get into a lot of detail here, in part because we're still going through these data, and I didn't want to tell you something that turns out to be not true, but also because I think you'll get the idea from just a few simple little bits of the data. So let me tell you a little bit about the cleanout part. This was chosen, as I say, as a means of depleting biomass, but it's meant to be analogous to a natural phenomenon, a disturbance in river ecology that has focused on the role of flash floods in purging systems and allowing reconstitution, elimination of invasive species, so plants that come up, invasive species along the banks of rivers. In this particular desert river outside of Phoenix, there have been some really classic ecological studies that show that at a frequency that is believed to be historically relevant, a flash flooding, this system recovers within a month. And it does so because there are plant and insect species that that replenish the system from the riverbed, the riverbed and the surrounding, I'm just blanking on the word, the surrounding ecosystem right adjacent to the river. Okay, so that's the basic finding of these studies. We said, hey, this actually has a natural analogy to what the human microbiome has seen ever since the very beginning, which is periodic diarrheal illness. Except that diarrhea comes in many different forms, and this is now after lunch, but I mean, there are inflammatory forms of diarrhea and watery forms of diarrhea, often because of pathogens or chemicals. They each have different kinds of timelines and consequences for that ecosystem. We were trying to mimic the watery forms of diarrhea without the obvious pathogen factors that might promote certain kinds of recovery patterns. But anyway, so that's what we did. That was the Cleanout thing. And here I'll just show you 8 people briefly in a little more detail. So these are their 16S-based profiles on an ordination space where the closer together they are, the more similar is the overall composition of those samples in those gut communities. And they're faceted by subject, 8 subjects. What you see is that they're all relatively tightly clustered despite our using the 2 axes that are meant to pull apart these samples as much as one can with this kind of analysis. So there's a fair bit of stability both before and after. The 3 days immediately after the Cleanout, they're colored in red. And those are the samples that you see a little bit further from the main clustering, in most cases, not all. Some of these people actually had trouble pooping for a couple days, so they actually didn't have a sample in the 3 days, or had only one in the 3 days after the purge. But you can see this now displayed in the bottom set of facets against time. Axis 1 is the y-axis. And you can see a change in composition, so upward increased value on axis 1 at that day 0 time point for some of these people. And we looked carefully at what are the organisms that seem to be most distinct at the time of disturbance. And this is one approach for doing that sort of thing. And again, this is published, but so not to belabor, organisms that are depleted at the time of disturbance and take a longer time to recovery are largely ruminococci. And they are organisms that turn out to be slow growers, dependent upon food particles for their livelihood in the gut. They kind of live around food particles. They're fiber fermenters. And you can easily imagine where a Cleanout has now eliminated their preferred little niche, and they're slow in coming back, but they do come back. It just takes 5 days instead of 1. Meanwhile, down on the other side of this scoring system, we've got Bacteroides, which are fast growers. They were back within a day. So they grow fast, but they also are very versatile, and they're happy with mucus. So if all the luminal stuff is gone, they can feed off of mucus-based polysaccharides. So that was sort of one little story that came out of this otherwise fairly discrete and transient disturbance. And we now have many more that we're looking at. The other one I just want to show just very briefly is the more selective depletion with an antibiotic. And there are many analogies one could choose. This is selective clear cutting I chose, but there are probably better analogies here. And this is what happened with the Cipro. And now I'm showing you all 60 people. There is a fairly hefty shift in, this is now taxon-based composition, and then there's a reestablishment of a stable state between 20 and 40 days after the 5 days of Cipro, and that's about 10 to 20 replacement times for the gut, if you're thinking of it as a kind of a continuous flow bioreactor. And these stable states appear to be different, at least based on taxonomic composition, than were the states prior. So this is kind of bad news, even for a relatively discrete antibiotic. If you think that this is the the measurement that matters most. These people were, by and large, asymptomatic. So we didn't even get antibiotic-associated diarrhea with these 5 days. We chose people that had not had an antibiotic for at least 6 months prior. That was part of the deliberate screening. And there's some interesting things. We're just, again, beginning to look at these data. There are organisms that behave similarly across the thousands that we can measure in these 60 individuals. Here are some of the clusters. And interestingly, I've got the cluster compositions here. You can't read them, but I thought I'd point out one. Faecalibacterium, a really common, highly abundant short-chain fatty acid producing organism believed to be important for colonic health. And if you were to look at cluster 1 only, where there are some Faecalibacterium strains, you'd say, hey, not too bad. They're back, more or less. If you look at cluster 3, there's some other Faecalibacterium strains. These are all Faecalibacterium prausnitzii strains, and those seem to be lost. So, one thing, if you were looking for stories to tell, you'd go back to all the ecological literature that says, hey, redundancy is good. Maybe these strains are doing similar things. We can reconstruct their genomes and ask if they are. And if the other ones that behave differently are not, at least competent to do same things. But this might support redundancy as a good thing. If only our political system would buy into this idea of insurance and so forth. The other thing is that there's a huge amount of individuality to how these subjects responded to the same exact disturbance. These are some of the most individualized responsive taxa as facets, and the lines are subjects. So you can see, different patterns among different individuals for the same strain, let's say, or species. Clearly, the whole thing is complicated. And then I just want to make sure that I haven't suggested that the only lens through which we should be looking at the microbiome is taxonomic composition, because we have metabolite data on subsubjects. This is a subject that actually had a diet disturbance and a Cipro disturbance. And I can walk you through this, but suffice to say, on the left is the diet disturbance from a metabolite point of view. These are fecal metabolites. On the right is the Cipro disturbance from the metabolite point of view. And what's confusing here, and I need to figure out, is how to color these so that I can distinguish more easily for you what is post-diet and what is post-Cipro. But in each case, it's red, which is post-disturbance. So here in the diet, you've got blue, which is all prior to the diet disturbance. You've got now samples move over here, very different metabolite picture. And they're there for just a short period of time. And then they go back. These red ones are post-diet. So they go back to roughly the same place. There's actually pretty good recovery of metabolites for a diet disturbance. But here on the right, when the Cipro profiles come over here, which they do, same kind of metabolite disturbance for Cipro, oddly enough, they now go over to here. All of the post-Cipro samples are over here in a very different state. So this corroborates, this goes along with the taxonomic composition data. Things have really shifted now from 2 points of view, and we're looking at others. Okay, a little bit about disease. Again, every one of these is a really interesting story with some data, some interesting early studies, mostly in animals where there's a good model. Some of these, there is no good model, so observations in humans. We've done some of these. I won't really focus on any unless we have time, you want to talk about them. But in each case, there is a reasonable case to be made to say, I see an altered microbiome at the time I see these diseases. I'm not sure whether one came first or the other, but in a few cases, there is some time series data to suggest that changes are beginning before overt signs of disease. So maybe there is a causal relationship, maybe there's not. If there is, we need to be talking about the community as the pathogen, not an individual organism. This is something that's a little bit hard for us clinicians to kind of get our minds around, in part because as an experimentalist, how are you going to prove that? So how do you do Koch's postulates on a unit of action, a unit of pathology that's hard to standardize, purify, and then not even have a model into which you can introduce it and give a reliable reproduction of the disease or not. People use germ-free animals. A long discussion, I think, could be had. They're useful for some kinds of phenotypes, but not for others. Germ-free animals start as aberrant things postnatally. And when you put a community in postnatally, you may recover some features, but you're never going to recover others. And so I think there's more to be done to work out how do we go about sorting these questions out. Cause or effect. If it's causal, is it only initiating a disease process, but then other things reinforce it, or is it a reinforcing factor? Other things initiate, like genetics or some untoward external factor like an antibiotic or an exposure to a pathogen, but now the microbiome adapts and reinforces the pathology. So there are lots of little scenarios here. How are we going to address some of these from a practical point of view? I know that Discovery Park is about making real some of the insights and discoveries and kind of applying them in useful ways, and that's what everyone wants to do with microbiomes, but it's hard. It's hard to do these very simple sounding things, either promote a beneficial one or restore a beneficial one that's been lost. And there are a lot of approaches that have been talked about, either introduction of potential community members or would-be community members alone or as small groups, defined groups, or even as undefined microbiota samples from elsewhere, and or the provision of key nutrients that favor what we think are beneficial members or disfavor the adverse members. Lots of possibilities. And then you've got the third factor, the environment, which I think is going to have to be addressed at the same time because no one of these things will actually work, it seems, from recent studies. And that is maybe correct an environmental problem that is part of this reinforcing function of inflamed gut, for example. Do we need to address inflammation per se, and or at least some component of the host response that will give the new community and its nutrients a chance to take hold? Otherwise, you just end up lapsing back into that previous valley of adverse state. How much of this will have to be tailored, we don't know. But the capabilities for engineering strains and really refining the kinds of reagents is pretty amazing. And here's where, again, the lessons from these small molecules may be really useful because each of them has a really interesting emerging story behind its normal properties and then the origins of its synthesis within the microbiome, and hence opportunities for deliberately producing or inhibiting these molecules. So let's take this one, indoxyl sulfate. Indoxyl sulfate is one of the most abundant so-called uremic solutes. It's a byproduct of a failed renal clearance system. When your kidneys go down, your bloodstream accumulates lots of products that turn out to be adverse or harmful in high concentration. Indoxyl sulfate was always there in health, but when your kidneys go down, it accumulates in high amounts, and it, among a handful of other small molecules, appear to be responsible for a lot of the pathology against blood vessels and against the kidney and heart that are typical of chronic kidney disease. And dialysis does not clear this thing. So it turns out that it is microbial in its earliest origins. And this is work that Justin Sonnenberg and Michael Fischbach at Stanford, my colleagues, sorted through in recent years. Turns out that it comes from the microbial metabolism of tryptophan to indole by an enzyme that they identified. It's a tryptophanase. It's made by a couple of common Bacteroides species, but not others. And then the indole is absorbed across the colonic wall, and now in the liver becomes indoxyl, actually even the colon wall, and then in the liver to indoxyl sulfate. So that's its history. It accumulates to really high levels, correlated with disease, and the question is, could you do something about it? And that's what this paper is about. I won't go through I actually pulled some slides on data here, but in short, what they showed is if you create a synthetic community that either has or has not Bacteroides species with this gene, you have dramatically different levels of indoxyl sulfate in the serum of mice, and that you can actually favor strains that make it or not by changing the diet in such a way as to favor these known Bacteroides species that make it, or disfavor them, and the reverse for those that don't. So this is all sort of demonstrated and possible, and now we have the tools for, selective expression, conditional expression with engineered promoters over, a million-fold expression ranges, suitable for lots now of these common commensals which had been genetically intractable until recent years. I mean, CRISPR makes some of this possible, and just good genetics and genetic engineering makes other parts of it possible. So in this paper, Justin shows that 6 different Bacteroides species can each be made to express a different fluorophore with a tunable promoter and reliably distinguished in spatial, structural studies of the distal gut microbiome in mice. Okay, so here's where I just want to mention, what does this all mean? What are the implications? Well, I spent all this time on the possibility of ameliorating disease and promoting health, but there's no reason why you couldn't just do things in reverse, why you couldn't make things that, or cause things to be made that are harmful and diminish things that could be beneficial. And I raise this only because there have been experiments in recent years done by well-meaning people that either inadvertently stumble upon an outcome that wasn't expected, where the outcome was exactly the opposite of what they intended. And there's also evidence that people, and some of our colleagues, are a little cavalier. And when an experiment sounds cool and possible, they do it first and ask questions later, like maybe should this have been done or not? And I could cite plenty of examples. But let me just pose this question. First of all, might it be possible for someone to do this, to actually increase the levels the way you'd have in chronic kidney disease without a failed kidney? And the answer is clearly yes. And what would be some of the consequences? Well, or the ways you would do it. You could certainly select for strains that are already there that make it. You could deploy engineered strains that make it in abundance. You could have them make it only when they see certain things, like tetracycline that someone needed for another intercurrent illness. And I'm not trying to deliberately be sinister here. I'm just saying that everything that we do and discover has the possibility for misuse. And in case you thought that there were only good, well-meaning people out there in the world, I'm really sorry, but there are people who are hell-bent on causing harm or being disruptive. And if we don't start thinking about this now, then others will, and we'll be behind the curve. And I think the question is, so what do you do when you start thinking that there are possibilities for misuse? Well, you might wonder, well, what are the likelihoods and what are the consequences? And that's a hard thing to figure out, because likelihoods involve really small numbers with big standard deviations, and consequences are potentially big numbers also with big standard deviations. And they're hard to estimate. So all I would say is, on this question, how likely and with what consequence, if we say that this unlikely as it may seem, but important, dark scenario happens only when a capable person is the same as or is friends with or working with, knowingly or unknowingly, an ill-intended person. It's the overlap, of the two circles there. Is there overlap today? I think yes for some things, I think no for other things. Tunable promoters, well, requires institutional, reasonable science. But are there ever individuals and institutions that might be willing to be co-opted, even unwittingly? Maybe. And if you don't believe that, then I would say you probably ought to believe this, that whatever the likelihood of overlap is today, the likelihood tomorrow is probably greater, even if you assume that the right circle stays the same. Ill-intended people are always going to be there, let's say in the same numbers. Well, the capabilities in biology are expanding like gangbusters. Lower entry to participation, lower cost, more efficient, et cetera, et cetera. So, so then you have to ask, do we have responsibility for thinking about this and kind of thinking it through? Well, how do we try to minimize the chance of this? And are we responsible? That's the nature of this question. I say absolutely yes. And if you think that you're too busy or that others can do this better, I think you're really wrong, because others will do it for you in a much less well-informed way. And if you don't believe that today, then I don't know when you might think that to be true or not. But there's danger in our ignoring questions like this and showing that we're actually doing something. So then it comes down to, this is my last slide, how do we govern ourselves? And here's where I and others have looked at the way science is being done today and have really struggled to find good models for how we ought to either self-govern or allow ourselves to be governed by thinking, knowledgeable, learning institutions. We don't really have that. And if one of you have ideas for where you think you see it now or what you think it might look like or by whom, please let me know. But this is something a lot of people, I think, ought to be or maybe starting to sort of think about. The NIH has not figured this out, by the way, in case you were hoping. But they care. And they might even one day support work of this sort at good interdisciplinary places like Purdue. So this is my summary. We are, in large part microbial, and that's good. It's incredibly complex, interesting, still to be sorted out. Lots of opportunities for both insight into how complex systems work, but also in how you kind of work towards ensuring there be a beneficial ecosystem. And then the possibility of people misusing, either by oversight or by deliberate means, those insights. And we have to also think about that as well. Okay. So just a few thanks. Les Ethlefsen and Arti Patankar are two that have been sort of leading that resilience experimental work that I showed you a little bit about, but were aided by a lot of other people, including Susan Holmes and her group in statistics, and some work by Casey Wong in bioengineering at Stanford, other collaborators shown here and not shown here, funders, and finally you. Thank you so much for having me here.
Questions, David, we have a little gift for you for being our speaker in this distinguished lecture series. It's a nice little globe, and so
thank you so much.
Thank you, David. Questions?
In the back.
The state needs to return to the same place in terms of, even from a simple physical chemistry standpoint, there are multiple states which are stable, and those states can be functionally giving the right response. So is there a notion in the field that it has to be the same state? Is it good or bad if it's not the same state?
Yeah, thank you. Thank you for the question. Sure. So the question is, you've given the impression that a shift to an alternative state is necessarily a bad thing, if I can summarize. And is that really true? And of course, it is not true. There are multiple alternative states, all associated with complete health, as best we know. And that was probably a place where I wasn't as clear because of my focus on a taxonomic perspective and not reminding you all that what we really care about is not even any of the things we're measuring, but those benefits. And those are the hard things to measure. But we have every reason to think that for these, for these well-meaning volunteers, we've left them in a place that works fine. They certainly have no symptoms. We won't know if when they travel to Bangladesh and drink a lot of arsenic-containing groundwater, they won't now have a problem they wouldn't have had because they lost their arsenic, mitigating and transforming organisms, because we don't measure that. But there's absolutely every reason to think that there are many ways, many paths to health, and many healthy places to be. Yeah, and some may be better adapted for whatever is the lifestyle or set of circumstances.
Thanks for the fascinating talk. This is kind of a riff on Gaurav's question, but with all the talk of functional redundancy, or maybe it's better put, functional degeneracy among strains, I'm wondering, after you performed the disturbance experiments, did you find any role for consortial relationships in recovery, and kind of if-then kinds of relationships among organisms that governed kind of recovery to new stable states?
Right. So you heard the question. The answer is, although it's early, yes, there is a sense that there are certainly co-occurrence patterns that are clear at the time of a disturbance, which accompany either return or the assumption of a particular new state that may be good, may be bad, we don't know. Yeah, so there are a lot of coordinated changes, and in fact, predictors may well be a set of features, not just of different measurement types, but also of different strain types as well, or things that different strains make, which together define that feature that predicts, is associated with stability, let's say, or not. Yeah, I don't know if I answered it.
Question over here.
Yeah.
Do you see any increased incidence of diseases that occur after a complete schedule of, for example, ciprofloxacin? If you're given the antibiotic to get over an intestinal infection, and you take it for 2 days, you feel great, but you still have another 8 days that you take it because that's what the doctor prescribed. You obviously have a total turnover of the gut microbiome. And I've listened to some other people suggest that the microbiome might even influence your response to therapies for cancer and other things of this sort.
Yeah.
You didn't mention much about that.
Yeah.
But is there, when you do this total elimination of the current microbiome with a very extensive schedule of antibiotic, do you see any consequences to that people haven't examined to date?
Yeah, there's several questions in there. First of all, nothing that we have done clearly has totally eliminated anything. In fact, we probably haven't come close, nor have those who have used deliberate population, decolonization regimens in the clinic or on the farm. It's actually very hard to eliminate all that might be there. There are reservoirs that presumably reside within the boundary of the system, the way the desert river has and the way we have in our gut. And yes, there are consequences even with limited depopulations and depletions. And with antibiotics, you probably well know, there is, roughly speaking, a 15, 20, 25% rate of diarrhea, of antibiotic-associated diarrhea in people who are given more prolonged periods of antibiotics, but that's usually in the setting of a disease for which they needed or was thought to need the antibiotic. It gets complicated, but there are clearly overt adverse consequences of using things like antibiotics, overgrowth of organisms like Clostridium difficile, et cetera. But as for functional, people are now looking, there's a great paper by Bali Pulendran, who did this work at Emory. He's now at Stanford. He looked at the effect of antibiotics in human volunteers on their response to flu vaccine. Did you see this paper? It's in Cell a month ago maybe. And in short, what he found was if you give people, he gave them 3 antibiotics, trying to really knock down their microbiota, and right at the peak of the antibiotic, he gave them flu vaccine. If you had a history of annual flu vaccination, you still didn't really affect the antibody responses. But if you did not, if you were vaccine, there's nobody naive, but hadn't had vaccine for many years, so poor memory responses, and then you did this antibiotic vaccination, you had impaired antibody levels and kinds of antibody to flu vaccine. So you are doing something to your immune system when you take antibiotics, it seems. Not surprisingly.
Okay, last question.
Thank you very much for your talk. So I was curious about the paper you discussed with indoxyl sulfate and quickly looked up what they used as a dietary intervention, and it turned out to be fructans, which are used, well, they're the culprit in some people who have irritable bowel syndrome. They're fed, or they're told to follow a low fructan diet, and now that there's suggestion that there's a consequence for reducing your dietary fructans, is that something we need to start to consider, either in those patients starting to monitor their levels of these dairy mixed solubles?
So
or
no, I think
intervening?
it's an interesting question. First of all, these are mice,
Right.
and the reason that they use fructan is because these actually were germ-free mice repopulated with a very small defined community, which included a Bacteroides coccae, which does not have the tryptophanase, can't make the small molecule, and a Bacteroides, forget, maybe ovatus or something that does. And coccae loves this fructan, and ovatus, whoever it was, does not. And that was a simple way for them to tip the balance in favor of coccae and drop relative amounts of ovatus. And that was simply a maneuver to show that the predicted effect on indoxyl sulfate in the serum of these recolonized mice followed the expected pattern. That's all. What makes this really complicated is that, just as you say, each of these nutrients has particular uses and proclivities by different members of our microbiota. We each have different microbiotas, and the endpoint you might desire, it could be varying hugely depending on whether you have chronic kidney disease or you're malnourished or whatever, you have irritable bowel syndrome. So it's going to be scenario and person dependent, and the manipulation may have to be actually even more precise than a broad fructan of the sort they used. Sure.
Okay, let's thank David for a great talk.